Lactoferrin structure, function, and applications. I'm Dr. Martin Montes. I'm infectious diseases specialist and director of the immunology laboratory at the Tropical Medicine Institute next door with Dr. Teresa Ochoa. And she asked me, and I'm very thankful to be here with you and share directing this welcoming ceremony. So we're going to start with our keynote lecture, titled Lactoferrin, 80 Years and Counting, by Professor Hans Vogel. Professor Vogel is a professor of biochemistry at the Faculties of Science and Medicine at the University of Calgary in Canada. Dr. Vogel's research interests include the application of protein chemistry and molecular biology that are used together with high-resolution NMR spectroscopy and various biophysical methods. He has an active interest in studies of antimicrobial peptides and related host defense proteins such as lactoferrin, lysozyme, and lipocalin. Recent studies have focused on the antibiofilm, antifungal, and anti-cancer activities of various peptides. Please welcome Professor Hans.
Well, thank you very much for that very kind introduction, and it's a great pleasure for me to be able to talk to you tonight as the keynote speaker. Because today is a bit of a special day, or this year is a bit of a special year, because it's exactly 80 years ago that lactoferrin actually was described for the first time in the scientific literature. And so a lot has happened since that time. Now, when lactoferrin was originally discovered, it was discovered as a red-colored protein that was present in milkweed. And so that's part of the slide here. This is how lactoferrin was originally discovered. And it took a while until they realized that there were actually two iron atoms that were bound, or iron ions that were bound to the protein. And it doesn't bind by itself. There is actually an anion that has to go in there and bind together with the iron. And in most cases that's carbonate, because it's around us everywhere and it gets into all of our buffers even though we don't realize it. But there's several other anions that it can actually also bind to the lactoferrin. The first crystal structure was solved in the 80s, the late 80s. And you know, that showed a very interesting structure that I'm going to talk a little bit about so that you know a little bit more about the background of the protein. And after that, we have had a plethora of all kinds of physiological studies, including clinical trials of small sizes to reasonable sizes to decent sizes. And none of that, unfortunately, has led to any clinical implications yet. So where we are today is that if you go to Amazon.com, you can actually find 10 different lactoferrin preparations, all promising that you will be healthier, live longer, enjoy life more, etc., etc., etc. So obviously, lactoferrin does many, many wonderful things. So first of all, I want to sort of say a few words about the background to this meeting, because the background is something that I was not involved with, but Tony was involved with it, Tony Schreiber and Leonard was involved with it. And during the early days, the idea was that there is a lot of lactoferrin in human colostrum. And there's not a lot of lactoferrin when you actually look at baby formula. So maybe we can put more lactoferrin into the baby formula. And indeed, some of these ideas are starting to come to fruition. Some companies around the world, some of them are sponsors, so I should acknowledge their contributions to this meeting. They provide now products which have some extra lactoferrin. Whether that is good or not, we still don't really know, but it is something that is at least a good step forward in this process. Now, before I go on, I first have to thank one person in particular, and that is our own Iron Lady, Teresa Lactoferrina Ochoa. Without her, we wouldn't have a meeting here. So, on behalf of all of us, Teresa, we really appreciate all the effort that you and your staff have put into this, because so far it's already been a wonderful meeting, and the program for the next couple of days looks wonderful. So thanks very much. Now, as Bo Lunderdahl said this morning, he says, be careful taking your wife to a conference because she can be your worst critic. And that made me think of something that my wife said to me. She says, "You're going to Peru, you're going to talk about lactoferrin." She says, "I think you know something about lactoferrin, but what do you know about Peru?" And so I had to really think about this. And in fact, I didn't know very much about Peru. But when I was young and I was a student at the university, I should be studying for my exams, but instead I read all these comic books, and one of my favorite characters was Tintin. And Tintin, on one of his adventures, actually made it to Peru. Now, the Peruvians probably know what's going to happen in this particular case. Anybody wants to venture a guess as to what the outcome of this? Yes, yes, yes. The Peruvians all know what's going to happen. The llama or the alpaca spits. So this is what I know about Peru, is that watch out for llamas or alpacas. If you see them walking around here in the wild in Miraflores, don't stare them right in the face because they might spit at you. So this is a warning. Now apparently, the end to the story is that eventually Captain Haddock, who is the main character in this particular thing, actually manages to get back at the llamas. So there is a way to beat the llamas, but I'm not promoting that you try this in real life. All right, so let's go to lactoferrin now and get a bit more serious. So lactoferrin was originally discovered in 1939, and it was discovered as a protein in milk whey, and it took till about 1960 for the protein to be completely purified. And then the crystal structure clarified a lot about how the protein actually worked, and the crystal structure showed that lactoferrin was actually part of the transferrin family of iron-binding proteins. And that was a major discovery. People already had concluded that based on partial sequences that they knew for the protein. But when the first crystal structure came out, it was actually the crystal structure of lactoferrin came out before that of transferrin. It really set the stage for this whole field. Now, I'm already going to give you away part of my secrets here, that there is a major difference between transferrin and lactoferrin, and that's going to be part of the story of my talk. And that is that transferrin, which is a very well-developed biological function and action, has a very different role in biology than lactoferrin has. And most of the differences come from this extremely highly positively charged N-terminal domain of lactoferrin. And I'll get back to that later, and I'll explain to you what I actually mean by that. Now, before we go that, in 1939, there was actually a meeting held. Batteries running out already, I think. So in 1939, there was a lactoferrin meeting held, but there was only one participant. And this was his name, because he was the only one in the world that knew that this protein existed. And this is actually a very interesting character, because he comes from the same country as Vogel, right? So you can properly pronounce this name. And it's also, unfortunately, the same year that he passed away. And this was his wife. So it was a husband and wife team. They worked at the Carlsberg Laboratories, which was actually a very famous laboratory for biochemistry in those days. Many important discovery in the early days of biochemistry came from the Carlsberg Laboratories. And Søren Peter Laurits Sørensen, he actually also was known to be the first to come up with the concept of pH. So this man has made two very important contributions to the world. We all talk about pH. We think it's a normal concept these days, but he introduced it originally. And he was also the first discoverer of the red protein in milk whey. And that's what we're all interested in when we come to this meeting. And so this initial discovery led to about 8,500 papers that you can now find in PubMed that actually talk about lactoferrin. So where is lactoferrin found? Just doing some technical corrections here. So lactoferrin is actually found very highly in milk colostrum, and it goes off over time as the infant is being fed by the mother. Another place where the lactoferrin concentration is unbelievably high is actually in human tear fluid. 80% of the protein in human tear fluid is made up by 3 proteins in about equal proportions: lactoferrin, lysozyme, and lipocalin, tear lipocalin, a unique lipocalin. So tear fluid is a rather remarkable fluid. It's made here in the lacrimal gland. It runs over your eye and then it drains into your nose. So it's constantly being replenished. But lactoferrin plays obviously a very important role there because there's so much of it present. Another place where there's a lot of lactoferrin present is in the neutrophils. So these are the white blood cells that go around, they police everything. They're the kamikazes that try to kill everybody who they don't want to see. And they do this in part through these storage granules that, in this case, this particular storage granule is completely full again with lactoferrin, and there's this friend, lysozyme. And so they often occur together, lactoferrin and lysozyme. Now there's many other bodily secretions where you also find lactoferrin in pretty decent concentrations: saliva, sweat, airways, various fluids that the body secretes. And so they can also create lactoferrin in pretty high concentrations. All right, so if we now look at the function of lactoferrin, lactoferrin is an iron-binding protein. Now, I was originally trained as a chemist, so I actually know the names of all these elements in the periodic table. There's another person in this room who knows the name of many of these elements in the periodic table, Boer Leonardal. He actually even knows how much zinc and iron and copper you should put in a baby's diet in order to look and make them feel healthy. But I know that for many of you, you don't really care about all these other metal ions, so there is a very simplified version here of the periodic table of elements, which is called the iron-centric perspective of the universe. And you can see that here. Iron is clearly the most important and the only important relevant metal ion in the whole periodic table. And so the only thing you have to know about iron is there's actually 2 forms of iron in our bodies and in nature. Iron 3+ and iron 2+, right? And they go by different names. One of them is called ferric iron, and the other one is called ferrous iron. So that's iron 3+ and iron 2+. And lactoferrin selectively binds iron 3+. And why is that important? Because that's the form of iron that exists in an oxygenated environment. That's outside of the cells, that's in our body, in the circulatory system. That's where iron 3+ is present. Inside the cells, we have iron 2+, ferrous iron. That is not particularly good for lactoferrin. Lactoferrin does not like to bind iron 2+. So clearly, this protein is designed for functions originally outside of the cell where it can interact with the iron 3+. All right, so this is the first crystal structure that was published by Ted Baker. And many of you who've come to these meetings regularly over the years will remember Ted Baker. He was part of our International Scientific Committee until he turned 70 years old and decided that life near to the beach in Auckland, New Zealand, was too attractive to travel the world, and so he doesn't come to these meetings anymore. But he and his team, including his wife Heather, published this structure. And you can see it's an interesting structure because if you look at it, there's some symmetry to it, right? You see here, there's iron sitting at the bottom of a very deep cleft between this yellow domain, which is the first end domain, and this green domain, which is the second end domain. But if you then look at the other half of the protein, you see there's another C1 domain and another C2 domain, and again, in the cleft there at the bottom, that's where the iron 3+ actually binds. And actually, there's some sort of gene duplication that has taken place in order to bring this one form, because this form is structurally homologous to this form. Also, the sequence of this is some homology to this. So there's some gene duplication event that has taken place in order to create this protein and make it a protein of 80 kilodalton. Now, why is that important? Why would you care that a protein becomes bigger from 40 kilodalton to 80 kilodalton? The reason is the kidneys. Because the kidneys clear small proteins, right? And in this case, you want this protein to stay around. It's the same for transferrin, it's the same for lactoferrin. These proteins are designed so that they can stay in the circulatory system. You see this binding of this iron 3 plus occurs at the bottom of this very deep cleft. Now, what happens to this protein if the iron falls out or is taken out of the protein? So here you see the protein that we just saw in the previous slide. See the two irons are in on both sides, and here you see the iron has been removed. And what has happened is the protein normally closes on that iron, but now it completely opens up. So the structure opens up quite dramatically. And people call this often the venous flytrap motion in order to sort of describe the venous flytrap plants that like to close on their targets, the insects that land on the venous flytrap plant. And so this is a very famous sort of motion that is seen in this particular group of proteins, the transferrins and the lactoferrins. And you see it occurs, it's hard to see, it's easy to see for this particular domain, but the same motions occur in that particular domain on the back. Now if you look closely at the iron binding site, this is what you see. The iron binding site is actually made up of two tyrosine side chains, a histidine side chain, and an aspartate side chain. That's 4 ligands. Iron 3 plus prefers to have 6 ligands. So in order to make this into a very strong iron binder, you have to do something else. And this little thingy here, that's the carbonate. That's the carbonate that we pick up from the circulatory system to make up the total of 6 ligands that we need to make a very strong iron binder. Without that carbonate, the iron is bound very, very weakly because you only have an incomplete coordination site. That's what the inorganic chemists call it. And so this carbonate is crucially important to the whole iron binding equation. All right, now, what else can bind in there? It turns out there's lots of things that can happen to this protein because protein actually turns out to be quite nonspecific. It doesn't just bind iron 3 plus, but it will actually gobble up most iron metal ions that have a 3 plus charge. And that includes things such as aluminum 3 plus, scandium, gallium, thallium, bismuth. I'm just showing off my knowledge here of the periodic table. This will be on the exam, by the way, at the end of the lecture. The reason that that is important is that they all have different ionic radii. So there is actually quite a change in going from the size of aluminum to bismuth. Bismuth is almost twice as large as aluminum. Aluminum is slightly smaller than iron 3 plus, whereas most of these other ones are the same size or larger than iron.
Yo estoy grabando, puedes ponerte.
Fit in there. There's also a few divalent ions that can actually fit in place of that particular iron. Now, instead of the iron, of the carbonate, you can also put in something that's called oxalate. Now, oxalate, you say, who cares? But a lot of medical doctors care because calcium oxalate is what gives you kidney stones. So oxalate actually occurs in our food. We ingest it regularly, and so this is a reason that this is important. When iron oxalate binds to lactoferrin or any of the transferrins, it's impossible to get that iron out. You can only get the iron out when the carbonate is in there, and I'll show you in a minute how this is done in the case of the transferrins. So I'm showing you this little diagram. This is from a paper that we published a while ago. I'm an NMR spectroscopist. You heard that in the introduction, so we've been doing lots of NMR in this group of proteins. And we made use of the fact that thallium has good NMR properties. You can label the carbonate with carbon-13. You can label the oxalate with carbon-13. And from this we can conclude how one side fills after the other. We can also see that the carbonate actually links directly to the metal ion. And we could see that this oxalate bound in a different way than the carbonate is. And we predicted that it was in what we call a 1-2 bidentate manner. And so we were very happy when 5 years later, T.P. Singh actually published a crystal structure for the active site of, in this case, force, diferric, lactoferrin, and you see this is for the C lobe, and this is for the N lobe of the protein. You see the tyrosines are there, the histidine, the aspartate, but there sits the oxalate in both cases. And if you look at the coordination, you can see that the oxalate, the oxygens of the oxalate, which are these two, actually are pointing towards the metal line, whereas in carbonate it would be those two that directly coordinate to the metal line. Now, why is this important? Because if you eat too much spinach or too much rhubarb, you end up with a lot of oxalate in your system. And so the oxalate can actually offset your iron metabolism to some extent. So you want to be Popeye the Sailor Man, be careful, don't eat too much spinach, because it could be harmful to you because of the oxalate content in there, apart from the fact that you can get kidney stones from the oxalate. Anyway, we can talk over this over a beer or wine tonight, because I think it is something that we normally don't talk about at this meeting. All right, now, people knew what transferrin was doing, so I'm going to review that with you very briefly, because transferrin is enormously important in our bodies. And so I'm deviating here a bit from lactoferrin. So where does the iron come into our body? It comes through the gut, and of course from our food, and we take up about 2 milligrams of iron per day from our food. That goes into the plasma transferrin pool. There's only a small amount of it, about 3 milligrams. Now, a person of 70 kilograms, so somebody a little smaller than me and a little bigger than Teresa, somewhere between there, somebody like that has about 5 grams of iron in their body. Where is most of that iron? That iron is actually mostly in the erythrocytes, in the red blood cells. The hemoglobin is loaded with heme, with iron in it, so that it can carry the oxygen through your body. The erythrocytes, though, get recycled by the macrophages. The macrophages store and keep the iron and release it again to replenish the plasma transferrin. The bone marrow, of course, makes the erythrocytes, so this is part of the cycle. The buffer is actually in the liver. There's about 1 gram of iron sitting in the liver, and there's another 300 milligram or so sitting in the muscle tissue. Now, plasma transferrin is the job of transporting iron between all these different tissues. And so it has the job of also getting into the cell, because it's got to bring the iron into the cell. And so how does it do that? It does that through so-called transferrin receptors. And this is a picture of a transferrin receptor in action. Here you see a cell, say a liver cell, a hepatocyte, and here you see a dimeric transferrin receptor, and in comes this holotransferrin. So that's the iron-loaded form of the transferrin, and it binds. It only recognizes the iron-loaded form. Then you form these clathrin-coated pits, you form these endosomes, and now you have to get the iron out of the transferrin, because it sits in there extremely strongly. And the cell has figured out a very simple trick to do this. It starts to pump down the pH in here. And what that does is it protonates the carbonate. And the carbonate now no longer likes to sit on the iron, and it falls off, and now you can easily get the iron out. Right? And so this is a very neat trick in which that synergistic anion of carbonate is essential for getting the iron out of transferrin, simply by lowering the pH in this particular vacuolar system. The cell is very efficient because it returns the receptor and the apotransferrin to the surface, and it gets recycled and you can use it again. So it's a very good system. The cell doesn't like the apotransferrin, or the transferrin receptor and the apotransferrin don't bind to each other. They only bind when they're iron loaded. And here's a picture. Unfortunately, this was a movie that was wiggling, but sorry, I couldn't get it to work this afternoon. So you won't see the wiggling proteins there. But this is a complex of the transferrin receptor and the transferrin. So when people started to think about what does lactoferrin do, they were originally biased by thinking about what does transferrin do, right? And so they said, well, okay, there must be a receptor like this for lactoferrin. And there isn't. There isn't. That's basically what it comes down to. There is no receptor like this that works for lactoferrin. And so then the question becomes, what can you do? And now I'm introducing Professor Bo Lunnerdahl, one of the members for our international committee, whose team has done a lot of work in this particular area. And they have described a number of receptors for lactoferrin, and of course they started to study in the intestine. Why? Because when an infant drinks milk, what is the first place that the lactoferrin comes in? It comes into the intestine, and maybe from there you can take it into the body and transport it further. Now the so-called now intellectin receptor binds the lactoferrin, and then it is targeted to the nucleus. So that's already quite different than what we saw with transferrin. In the case of transferrin, it was recycled back out. It just went through the endosome to get the iron out, and then it was recycled. In this case, it targets to the nucleus. I cite this directly from his paper. One of his other papers showed, and this is what was disheartening at the time, was that it didn't matter if the iron was bound to the protein or not. So this now makes it totally unclear what the role is of these receptors in iron transport from lactoferrin to the infant. And we still don't have a very clear picture of whether lactoferrin actually is essential for bringing iron to the infant. We think it does, we think it is important, but the current biochemical evidence that we have does not strongly support such a role. Now, if that is not the main role for lactoferrin, then what is the main role for lactoferrin? Right? And this is where this conference spends most of its time. We talk about all the other activities that this protein does. Right? One of them, which still all hunkers back to the days of iron binding, is that, yeah, you can withhold iron. Lactoferrin actually binds iron better than transferrin. And so if you pull the iron out of the system, the bacteria cannot get to it because the bacteria, just like we do, they need iron to grow and to proliferate. But another thing that people discovered quite early, and this is really interesting, is that they found that human and bovine lactoferrin can do the same thing, can actually bind endotoxin or LPS. And that is essential because what that means is it can actually detoxify the LPS so that we don't trigger things leading towards sepsis, because the endotoxin reaction can trigger parts of the immune system, and when that goes too far, we may end up in septic shock. So this is a very dangerous sort of reaction, and lactoferrin is capable of modulating that interaction. Lactoferrin can do other things in the immune system. It can trigger various immune cells. I mentioned here the activation of dendritic cells, but there are many other immune cells that have receptors that can react to the presence of lactoferrin, and so things can get done. The other thing that people found is that you can actually generate a group of antimicrobial peptides from lactoferrin, from that N-terminal highly charged positive region, which distinguishes lactoferrin from transferrin. And there's other activities, antiviral, antifungal, anti-cancer, and they probably all relate to that same highly basic region. All right, so here we have a group of host defense peptides. Lactoferrin is, in this case, the orange one, and it can do two things. It can have a direct action. It can actually act on bacteria directly and disarm them, but it can also have indirect actions where it acts through various systems in the immune cell. And so these are two different classes of action, and unfortunately in the literature people don't always distinguish enough between these two, so in some papers it's not totally clear what the actual action or mode of action of the protein is. Now this is a slide we've already seen several times today, so I'm not going to belabor it, but there's way too many things that lactoferrin does. And we still don't really know what the essential things are that lactoferrin actually can do. And this is from a paper written by a scientist. But it's not just the scientists that have caught on to this, it is the whole popular community that is into orthomolecular health that is also caught into this. And I just was surveying the internet for some inspiration for this talk, and I came across this website, Self-Act, where somebody actually came up with an article that's very recent, 20 health benefits of lactoferrin. And basically it's all the things we talk about at this meeting, and then a nice diagram, and then of course a link to Amazon.ca or Amazon.Peru or Amazon. Wherever you live, in order to start ordering your favorite bottle of lactoferrin. So this is where the health community has gone with this particular protein. And so the onus is on us, to a certain extent, to make sure that we only see the healthy effects happening when this happens. Unfortunately, in all the clinical trials that have been done today, nobody's ever reported that ingesting bovine lactoferrin is not good for you. You might get diarrhea, but we all recover from that after some time. But, bovine lactoferrin is, in general, considered to be totally safe, even at high concentrations. So even though maybe this is a practice to be frowned upon, it is also a case that it probably doesn't harm. So here's the famous blue region. So this is the positively charged region. So this is the protein structure, the same as this. Here you see the secondary structure, the backbone, how it's folded. Here you see the surface of the protein. And the blue regions is where all the positive charges are. And you see this is extremely positively charged. This is N-terminal region, this is bovine, this is human. And here is rabbit transfer. And you can look at the numbers. You can immediately see that this is a very basic protein, 9.11 isoelectric points, 9.22 isoelectric points, so basically completely blue. And then here, this is a much more normal protein, even though there is some blue, there's also some red to compensate for it. And so this lacks this totally positively charged region. So this is an intrinsic and unique property of lactoferrin compared to the transferrins. And so what does this do? Well, it binds to all kinds of things. It binds, I already said this, to the endotoxins, but it binds to glycosaminoglycans. It binds to polysialic acid. We heard about that today. And it also binds to DNA. And why is that important? Well, in some cases, we have this net formed by the neutrophils, and then it has to bind to DNA to stay close. But also, it can enter the nucleus, and then it can start to act as a transcription factor. So there's another reason why it likes to bind to the DNA. So this is a paper, one of my favorite papers in the field, from Dominique Legrand, who used to come to these meetings very regularly. And Dominique showed in here two important things. One, that if you bind LPS, you can actually overcome that. You can see that here. When you add lactoferrin, you can actually see that you can actually overcome part of the expression of this particular cytokine by adding more and more lactoferrin. The plot runs in this direction, so this is where the most lactoferrin was added. So clearly, this is the fact that you can suppress the immune system because the activation that normally goes through LPS and the toll-like receptor and all that is inhibited. But what he also showed is he actually puts this particular chemokine, interleukin 8, he put it on the surface, and then he watched it with serum transferrin. You see nothing happens, and then he watched it lactoferrin, and almost all of it comes up. So they're competing for each other on the surface of glycosaminoglycans. This is a haparin control, and it's not anywhere near as effective as lactoferrin displacing the ILA. So clearly, lactoferrin can interfere in all kinds of aspects of the immune system, and sometimes in a totally unpredictable way, because binding of IL-8 and all these other chemokines to the glycosaminoglycans helps in the presentation towards the chemokine receptors and triggering the immune cells. In this way, you knock it off, and it can no longer carry out this function. Now here's one of these neutrophil extracellular traps. We've already seen those before today. And they're the DNA that's extruded by the neutrophils. And you see there's a bunch of bacteria that get trapped in there. This is a sort of last-minute effort by the neutrophils to get rid of infections. So neutrophils play a very important role in this. Neutrophils can degranulate and release lactoferrin, they can phagocytose bacteria and merge it with one of these granules. So now lactoferrin and lysozyme connect together on here. And then the third thing they can do is they can actually mix one of these with the nucleus and then extrude their DNA with everything bound to it. And now you can trap the bugs in there, the bacteria, and now these proteins, including the histones, now can act on the bugs. Histones are actually quite antimicrobial proteins, not just lactoferrin, but histones are also quite antimicrobial proteins. So this is another interesting reaction. Now there's a large number of proteins that can bind to lactoferrin as well. And we can quickly go over some of these, but one of them is osteopontin, the other one's lysozyme, ceruloplasmin, which is important because it interacts with ferroportin, and then there's calmodulin, which is an intracellular protein. And there may be more. I haven't done an exhaustive search of the literature. Now, Dr. Leonardal already talked this morning about osteopontin, so I'll be very brief. Osteopontin is a protein that's present in milk at very high concentrations. It interacts directly with lactoferrin, a highly negatively charged protein. It's a completely unfolded protein. It has an extended structure. There's various forms, important also in bone remodeling. What we showed in our work, which is now 10 years old, is that we can actually bind 3 of these lactoferrins to one of these extended osteopontins. And what we concluded in our paper was the concentrations of lactoferrin and osteopontin were so high that this complex must always be present in a human. And then we suggested that maybe it protected lactoferrin from proteolysis in the gut because it was known at the time that osteopontin was not degraded, so the complex would protect it. And indeed, Dr. Leonardal, this is one of his publications on this topic that just recently came out in the literature, confirmed that protection of lactoferrin in the gut environment by osteopontin plays a significant role. Why that is important, we don't understand. But we know that these 2 proteins now, both majorly important in human milk, they are expressed in a coordinated ratio and they play this role. Now this is the only role because other people have actually reported that there are negative things coming from this osteopontin. In this case, this is a group of Swedish researchers from the University of Lund. They actually found that osteopontin can bind to lactoferrin, lysozyme, beta-defensin, and other things that the immune system normally uses, part of the innate immune system, and it inactivates them. So what is the reason for this? Here's another reaction where a protein in milk comes in and can do all kinds of weird things in the immune system that you wouldn't necessarily want to happen in this case. So this is still totally unclear as to what the relevance of this is. So there's a lot more to be learned about the role of osteopontin. Now I want to say a few things about lysozyme because lysozyme and lactoferrin occur often together in the same biological compartment. We find them in neutrophils together, we find them in tear fluid together, and in both cases walking in high concentrations. Actually, lysozyme, most people don't know this, was actually discovered originally by Sir Alexander Fleming before he actually discovered the antibiotics. It's published in 1923, he discovered this in 1922. So this is probably the first ever antimicrobial agent that was described in scientific literature. It is an observation of some note. It's a small protein, it's an enzyme, and it hydrolyzes peptidoglycan. And why is that important? Because this is a bacterial cell wall component. We don't have peptidoglycan in our bodies. Bacteria do. So this enzyme is designed to go after bacteria and to hydrolyze their peptidoglycan. So it's an antibacterial enzyme. And, again, super high in human milk, together with lactoferrin. But this is a positively charged protein, so you wouldn't expect these 2 to interact with each other. But there's this very intriguing paper out in the literature, been out there for a long time, where they show that lactoferrin and lysozyme actually have a synergistic action, killing bacteria. So the 2 promote each other's activities. So we've been thinking about this for a long time, and without showing you the data, we've actually shown that the 2 can form a complex, And you can see that here. This is the yellow is the little lysozyme, the green is the bigger lactoferrin, and you see what happens. This is twisted around here. So this is the end lobe of lactoferrin, very positively charged region, and here sits this lysozyme. And if you look at this from the top, you see how blue this whole surface becomes. This is a totally positively charged region. This has become twice as large by sticking the lysozyme onto the lactoferrin. Why is that important? Because the surfaces of bacterial cells are negative. So this protein complex will dive onto the negatively charged bacterial surface and will start to do things to it. And one of the things that lactoferrin does very well, it actually punches holes in the outer membrane of gram-negative bacteria. And in doing so, it makes holes for the lysozyme to go in, because lysozyme by itself cannot go in. Lysozyme actually wants to go after this peptidoglycan. This peptidoglycan sits between the outer and the inner membrane, in the case of gram-negative bacteria, and lysozyme needs to get there in order to chew this up. Because if you chew that up, the outer membrane will actually disappear, and the cells become unstable and they no longer survive. So that's why these two proteins like to work together. Now, I just want to say one word about my friend Vadim Vasiliev, who is pictured in this slide, also a member of the International Committee. And this is one of his papers, and this paper has thrown me for a loop for a long time, because what he shows here is that lactoferrin binds much better than transferrin. Well, that's been the tone of his talk. And so this makes perhaps good sense. Must be the N-terminal positively charged region again, blah, 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 blah. But the function of ceruloplasmin is actually to deliver iron 3+ to transferrin. And so originally I had expected that transferrin would bind stronger because ferroportin brings the iron out of the cell in the form of iron 2+, ceruloplasmin kicks it up to iron 3+, and then it goes into the transferrin for transport through our body. And so this is a rather remarkable observation, and I still think there's more to be learned about this, because why lactoferrin is interfering in this process could actually try to tell us something about what happens under conditions where lactoferrin is present at high concentrations in the plasma. And finally, this is calmodulin inside the cell, binding to lactoferrin. This is work from our own group. It doesn't really matter so much, but again, lactoferrin binds, transferrin doesn't. And we concluded that after a lot of work that the calcium calmodulin may promote the uptake of lactoferrin into the nucleus, which is important because that's where lactoferrin can act as a transcription factor. All right, so now we get to another part of the talk. So the conclusions here are that ionic interactions normally play a dominant role, and it's always that highly basic and terminal domain of lactoferrin that is essential for the protein interactions, for the interactions with the glycosaminoglycans, and also for the intellectin receptor interactions. They all play in the same part of the molecule. Now, we should be done by now, but lactoferrin also has all these intriguing peptides. And this is what I would call the gang of four. This is harkening back to the Chinese Cultural Revolution. Right? But Mao Zedong is not very popular. Only older people will remember this. But these three people are all in the International Scientific Committee because they like to go after bacteria with peptides that come from lactoferrin. And this person here, Tony Schreiber, actually is interested in direct bacterial LF interactions. So Lydia and myself, we do a lot of work with peptides, and we also work a lot with our Chinese colleagues from the Beijing Institute. So the original discovery of these peptides actually comes from a milk company out of Japan, Morinaga. They were the ones who first described in 1992 already that you could actually make two peptides, one out of bovine lactoferrin and one out of human lactoferrin. Now, they made a little mistake because their methods weren't so good, so we actually corrected the sequence, and Håvard Jensen, as you can see, is the co-author on this paper, so you can attack him as much in it as you want to attack me. And by and large, basically, this one has drawn the most attention because this is the most potent antimicrobial. This one is not a particularly good antimicrobial. This one is an excellent LPS binder. And so my lab has spent 2 years trying to find an expression system in which we could make tons of this, because we thought if we could make this, we could mop up LPS wherever we wanted. We've never been able to make it. So we sort of given up on it. We don't talk about this anymore at this meeting, which is unfortunate, because I still think this is a very important reaction in the whole scheme of things that we have. So most of the attention has been on this guy here, and there's actually been a number of things that we can tell you about this. So here, this is the intact lactoferrin molecule. This is the N-terminal positively charged domain. And this green region is what's actually going to get cut out by pepsin. And this occurs in the stomach. So this is a naturally occurring product. It's not an artificial synthetic peptide. It's a naturally occurring peptide that can be made from bovine lactoferrin. And what happens is you see it has an alpha helix and a base strand that make up the structure of the protein. But when you take it out, it becomes a beta hairpin structure that is held together by the single disulfide bond. And for the biochemist in the audience, this is a completely amphipathic structure, meaning that all the hydrophobes are on one side of the peptide, they're all up here, and all the positive charges are on the other side of the peptide. And again, this loves the negatively charged bacterial membrane, because it's got all these positive charges. It dives on there, rotates around, sticks the hydrophobes in, the bacterial membrane is perturbed, and the bacteria die. So this is how a lot of antimicrobial peptides work. So this is interesting because you have a protein which in itself is a bit antimicrobial, but you take a peptide out of it and now it's 20 times more antimicrobial. So that's really quite a step up from small activity to really big activity. So that's why bovine lactoferrin has drawn so much attention. And people have tried to make smaller versions of it, but people have focused in on this particular region here where there's a bunch of tryptophans and arginines, and in my talk tomorrow, I will talk more about that because then we're talking about antibacterial effects. There's another region here which has drawn attention in the literature, which is this part here, which is really weird. This is the human lactoferrin structure. This is an 11-mer peptide which has a sequence with 4 arginines in it. That is a nuclear localization signal. And people think that this is why human lactoferrin likes to go into the nucleus once it makes it into the cell, because it has its own built-in nuclear localization signal. This is a lousy antimicrobial, this is a lousy antimicrobial, but this thing is very good at getting into cells. This thing is very good antimicrobial. All right, there's another one in the literature that was described originally by my friend Jan Bolscher from the Netherlands. He looked at this sequence of this protein. He says, you know, that purple region that lies right behind there also has some sort of positive charges and some hydrophobic things, so we can make an amphipathic structure. So we solve the structure for him, and it makes partially helical structure, part is unstructured. And this one also has some activity, but this one does not occur naturally. You have to make this one synthetically. And he also had the bright idea of actually sticking this together with the original lactoferrin. So here is part of the lactoferrin here. Here is the lactoferrin part that he put on there. He linked the two together. The structures show that there's some sort of flexibility here, but this part is helical, that part is helical. The problem with this is it's quite cytotoxic, which is quite normal for when you couple 2 antimicrobial peptides together. It's not a particular strike against this particular peptide. We've made dimer peptides, and usually what you see, the activity against the cell that you want to keep alive goes up as well. That's not a desired side effect. All right. Just want to talk for 2 minutes about the anti-cancer activities of this lactoferrin peptide, because this is also sort of an interesting study. The Norway group, Oystein Rekdal, they actually showed that this lactoferrin B peptide was a good anti-cancer peptide as well. And they dribbled it on cancer cells, and then they also looked at the topically transplanted tumors in mice, and they found that it was a potent anti-cancer agent. But he said that disulfide needed to be there in order to keep that structure intact in order to get that activity. The group in Canada, Dave Hoskin and Dalhousie University, he actually took that small 6 residue peptide from the protein and he tried to kill cancer cells with it, and it didn't work. So that's close. Yeah, okay. So he took this one, right, and he wanted to Oystein Rekdal used this whole thing, and Dave Hoskins says, can I do it with that one too? Make a long story short, he couldn't do it. But what he found was that if he put it in liposomes and delivered it right into the cell, it wiped out the cancer cells completely. So if you can get this thing into the cell, you can actually make potent anti-cancer. And we in ours have worked on that. We've done some work with anti-cancer peptides. This is some of our studies in this particular area. I'm not going to show you all these data, but here you can see this is lactoferrin. And you see as the concentration goes up, it has a potent anti-cancer effect against your gut cells in this case. This is a stable version of the lactoferrin where we replace the disulfide with something. All these small peptides don't work very well, so that agrees with the data of David Hoskins. And then these are some other antimicrobial peptides. And you might take a look at it and say, "Whoa, look at that one, that is always active." Well, that's melatonin, that's a peptide from bee venom. That's the most cytotoxic peptide on the planet. You put it on red blood cells, they explode. You put it on white blood cells, they explode. So this is completely distinguishing peptides, completely cytotoxic. And so you should never just look at these activities, but you should look at the specificities. And so here you see the specificity or the selectivity of this particular peptide. We measured what we got against the your gut cells and then compared it to what we get for normal white blood cells. And so if you keep the normal white blood cells alive, but you kill the cancer cells, now you've got something that you can start working from. And you see lactoferrin B actually has some of that activity. And if you look at melatonin, it has none in terms of selectivity, so it's not a particularly useful peptide. So this brings me to the end sort of my talk, because the other aspect of the whole lactoferrin field is all these clinical trials that people have been holding, and these are all different things that people have been trying to do. And I'm not going to say too much about this because it's A, not my area, and B, I wish I could say something super positive. I think that's a mild way of saying that we wish some of the results would have been a little different, and we're going to talk about this on Wednesday, because there's obviously something that we're missing when these trials are getting designed. And most of these have been done with bovine lactoferrin, and there's a number of people on our team and our committee that are active in this area, of course. Pierre Valenti, Teresa Lactoferrina Ochoa, and Dr. Tsuda from Nagoya. Now, there are some nice things in the literature. I mean, lactoferrin has finally reached the Lancet, although maybe not in the most positive light that we would have liked to see it there. There's other things. I just came across this in a journal called Critical Care, which is all the peoples in the ICU read. Iron metabolism is now becoming an emerging therapeutic target for critical illness. Lactoferrin plays an important role in this. This is an interesting peptide or paper in view of where we currently are. In low-income countries and middle-income countries, there are things to think about in terms of iron. And this is also an interesting paper I felt personally, because it might actually explain why lactoferrin is important without bringing iron to children. So, this brings me to my conclusions. And this is how I conclude the field. Lactoferrin is a multifunctional protein. That's all we can say at this point in time. I wish we could say a lot more about all its biological functions and what it does and how we can use it clinically. But you see that all the scientists working on this problem, they all have a different opinion on this matter. Now, of course, all scientists are blindfolded when they do their experiment. And so if you work on this part of the problem, you might think this is the answer. If you work on this part of the problem, you might think this is the answer. If you work on the tail, you think that this is the answer to the problem. And so we all have different answers while we're studying all the same thing. And so over beer and over wine tonight, we can talk about how we can maybe synchronize some of our opinions about this protein. And with that, I just want to briefly put up a slide with the people in my lab who work in my lab, and thank you for your attention.